should be good. It's just going to be 20 minutes.
Hi everyone, and welcome to today's webcast, Trends in the Feed Industry, Hot Topics and Advanced Technologies to Meet the Analytical Challenges of Feed Testing. Our first speaker today is Andre Schreiber from AB SCIEX. Andre is the Technical Marketing Manager for the Food and Environmental Testing applications, and he's going to provide an overview of some advancements in LCM SMS technology that will enable better performance for residue testing in feeds. Our second speaker is John Ruther from Eurofins. John is the Director of the Residue Testing Laboratory in Metairie, Louisiana, and he's going to discuss methods and applications offered by Eurofins to test for various contaminants, residues, and medications in feed samples. We're first going to start with Andre, so I'm going to hand it over and let Andre take it away.
Thank you very much, Lauren, and thanks to everybody for joining us today. Before I start off, I would like to just give you a quick overview about our company. AB SCIEX is part of the Danaher Corporation, and we are a global leader in developing best-class analytical techniques and tools, including LCM SMS instrumentation. And our instruments help scientists all around the world to answer important questions like what is in my sample, how much is in my sample, and how can I use that information to advance science, and in our case to make food safer and better. I would like to give a little overview about LCM SMS hardware and software. I would like to compare that a little bit to GCMS, compare different ion sources, different operation modes, to give you a big overview about LCMS and how it's used best in the application area of food testing, how we can get high quality quantitative results and good confidence in identifying compounds. And then I would like to address the challenge of matrix effects a little bit, how sensitivity can be used to minimize matrix effects as much as possible. And that's quite important because the matrix feed we want to cover today is one of the more dirtier matrices which food laboratories have to analyze. With this, quickly back to Lauren, and we want to answer some poll questions.
So we want to get a little bit of information about you, the audience. And so we have two questions, and you'll see them on your screen. The first question is, what type of mass spec instruments do you use in your laboratory? GCMS, GCM SMS, high resolution GCMS, LCMS, LCM SMS, high resolution LCMS, or you don't use mass spec? And the second question, what type of compound classes do you analyze for in your lab? Pesticides, antibiotics, veterinary drugs, steroids or hormones, natural toxins such as mycotoxins, chemical adulterants such as melamine, packaging migrants or processing contaminants such as phthalates and acrylamide, allergens, or vitamin and nutritional analysis. So I'll give another second to enter in your answers. Again, what type of mass spec instruments do you use in your lab, or what type of compound classes do you analyze for in your lab? And I'm going to stop the polls now. And it looks like, as far as the type of mass spec instruments that you're using in your labs, most of you are using LCM SMS with a mix of GCMS and some of the other mass spec systems, and a handful of people not using mass spec at all. And the type of compound classes looks like a mix between pesticides, antibiotics and veterinary drugs, and natural toxins as the leaders, with some others doing chemical adulterants and vitamins and nutritionals. Andre, do you have any comments or anything you want to say about the polling results?
Oh, very, very interesting distribution of people we have in the audience today. I'm glad to see that there are some LCMS users already, and of course also GCMS is used in most laboratories. But also very interesting to see that we have people in the audience who don't use mass spec at all, and that, I guess, will be a very interesting presentation for them. And if I look at the different type of compound classes and applications, yeah, that's a very typical mix what many laboratories are doing, and it doesn't come to a surprise to me that toxin, antibiotics, and pesticides are analyzed the most.
Great, thanks, and thanks to the audience for participating.
All right, let me start with a little overview on LCMS, and I try to compare it a little bit to GCMS because the people who are not familiar to LCMS yet they might find that comparison quite useful and interesting to learn from. So if we look at the typical setup of a GCMS instrument, it looks like this. So we have our sample introduction device, our autosampler. Then we have our GC with the column oven and the column. And then as the combining piece between the mass analyzer and our chromatography front end, we always have an ion source. That's always the very first part of the mass spectrometer. We have to turn our target compounds, the analytes we want to analyze, into ions first. And then we can separate these ions inside of the mass analyzer to differentiate different compounds. And the most commonly used ion source in GCMS analysis is electron impact. That's the gold standard, and single quadrupole mass analyzers are most commonly used for GCMS analysis. But also in the poll question, we could see that a number of laboratories are making the switch to GCMS MS analysis at the moment to gain sensitivity. If we compare this setup to an LCMS instrument, it looks very similar. We still have our autosampler, then we have our chromatography device, which is now based on liquid separation. And then we have our ion source, which is typically electrospray in the case of LCMS MS analysis. And only for a couple of compound classes, typically the lower polarity analytes, different ion sources like APCI or photospray is in use. The most commonly used mass analyzer for LCMS MS analysis is a triple quadrupole based instrument, but also there are a number of hybrids between different mass analyzers, and two very popular of them are the combination of triple quadrupole with ion trap, that would be a Q-trap, or the combination of quadrupole with time of flight instruments, that would be then like the triple top instrument. But you can see in general the system setup of GCMS and LCMS MS is very comparable, and the two big differentiating pieces are the ion source, electrospray in case of LCMS, and then the triple quadrupole setup for the mass analyzer. Let's have a closer look into these two pieces. Here's just a schematic how electrospray is working, and that is the most commonly used ion source. Probably 90% of all LCMS analysis is done with electrospray. We have our capillary coming out of the LC system, and we are spraying our LUNs containing the analytes we want to analyze through that capillary into the chamber of the ion source. And we typically use a little bit of nebulizing gas to make that spray process very reproducible. And then to the tip of that capillary, we apply a high voltage, typically a couple of kilovolts, and that is ionizing everything which is present inside of the droplet. And these ions try to be as far away from each other as possible, so they're typically located on the surface of these droplets. And while these droplets now flying towards the orifice of the mass analyzer, where the vacuum system of the instrument is starting, these droplets slowly evaporate. We typically apply some gentle heat to speed up the evaporation, and at the end of the evaporation process, there's something happening which is called Coulomb explosion. The droplet is exploding, and ions are released from this droplet. And now ions of a specific polarity, whether positive or negatively charged ions, go through electric fields into the mass analyzer region. If we look at the spectra generated with electrospray, we typically see that these spectra are dominated by the molecular ions. We typically see something like protonated ions or deprotonated ions, depending if we operate the instrument in negative or positive polarity. And it's the best ionization technique for all polar to medium polar analytes, which are analyzed by LCMS. LCMS. And probably biggest difference to electron impact spectra, if we compare it to GCMS, is that in GCMS we typically have way more fragment ions present in the spectrum because we apply a much higher voltage, 70 electron volt, which is generating fragmentation for these ions. And now I want to compare these two different type of analyzers. We have a single quadrupole instrument like a GCMS system, and we have a triple quadrupole instrument like an LCM SMS system. And the two most commonly used modes of operation for the single quad are called selected ion monitoring, and for the triple quadrupole instrument it's called multiple reaction monitoring. And we do that by using our quadrupoles of our quadrupole system to filter a specific target ion. In case of selected ion monitoring, we for instance filter the molecular ion of a specific analyte we are interested in. And if we use multiple reaction monitoring in LCMS, we filter the molecular ion, then we generate fragment ions out of this specific molecular ion, and then we filter a specific fragment ion for the target analyte again. And this double filtering is removing lots of noise from the chromatogram, and that's the reason why LCMS operated in multiple reaction monitoring offers such a high selectivity and sensitivity and became the gold standard for targeted quantitation over the last couple of years. Modern triple quadrupole based instrument can do that multiple reaction monitoring very, very fast. And that was very important for the success of a triple quadrupole based instruments for multi-component analysis. And typically also we monitor at least two transitions for each target analyte. And the ratio of these two ions is very, very characteristic for target analytes. And we can use the ratio for compound identification. Here's just an example of operating an LCMS instrument in selected ion monitoring mode and in MRM mode. Here a number of pesticides were spiked into a food sample and monitored, and you can easily see comparing these two chromatograms side by side that MRM is much more selective. We typically observe very little or maybe even no noise, and that gives us a much better signal to noise for quantitative analysis. And also MRM is much more sophisticated in handling data points, typically resulting in better accuracy and reproducibility for quantitation. One very key tool for a typical food assay is the ability to perform the scheduling of MRM transition, and that is it's shown here on that little schematic. In the acquisition method, we tell the software, the instrument, at what retention time a specific target analyte is eluting from the column, and then automatically the software is scheduling little windows to detect a specific analyte only around the retention time. And of course, that's much more efficient than detecting all the compounds all the time. This allows us to detect much more compounds, to speed up our chromatography, while not sacrificing data quality like accuracy and reproducibility in any way. And these schedule-based methods are used in almost every food application when many analytes have to be detected. Here's just an example of a typical multi-residue method for pesticide analysis. We used the scheduled MRM functionality I was just describing, and on top of that we also switched the polarity. We acquired negative and positive polarity in a single analysis, and the example chromatogram here shows about 250 pesticides, many of them in positive ionization mode, a couple of them in negative ionization mode. And the concentration you see here on the screen is 1 parts per billion. That is about 10 times lower than pesticide laboratories typically measure pesticides in food samples, and that allows us some dilution. And later I will explain what advantages you have if you have the ability to dilute your sample extracts. One quick example how it looks like if we apply that method for a real sample. So here we extracted some grapes using a catcher's procedure, which is a very common extraction protocol for residue analysis. Then we did some cleanup using PSA, and we diluted our sample extract before injecting it into the mass spectrometer. And the software was flagging automatically here the compounds which were present above 5 microgram per kilogram in our sample extract. And that data processing was done in our multi-quant software using the multi-component query. Here's a screenshot of that multi-component query. That's showing the result table for one specific analyte I filtered here. And at the bottom we have the peak review where we can look at chromatograms and maybe change the integration if necessary. The software was automatically flagging a number of samples where that specific pesticide was above the 5 parts per billion level we set in the software. They're highlighted in green. And the software automatically calculated the ratio of our quantifier and qualifier MIM and compared that ratio to our standard injection. And then we applied tolerance levels, and the software was highlighting that the MIM ratio confirmation was positive. And for that reason, the finding of carbendazim in these samples had to be reported. But sometimes the use of 2 MIM transitions per compound might not be sufficient to confidently identify a compound one, and then there are results where matrix can interfere with our signal of our target analyte, and then it's quite difficult to make the identification 100% right, and for that reason, sometimes more MS information is required to increase confidence of identification, and one way to do that would be to use a Q-trap instrument and use a mode which is called information-dependent acquisition. Here we have a method using our scheduled MIM survey we can use to quantify all the target analytes, hundreds of them in a single analysis. And we tell the software that whenever a specific signal goes above a threshold, that we quickly switch over to MS mode, collect a full-scan MS spectrum, and this full-scan MS spectrum can then be searched against a mass spectra library, pretty much like you would search a NIST library on your GCMS instrument, and this is significantly increasing the confidence of compound identification using the additional library functionality. And we did that last year when we found carbendazim in orange juice samples. So here you can see the MIM chromatograms of 2 orange juice samples on the left side, where we quantified carbendazim at 13 or 67 nanogram per milliliter. And to increase our confidence, we automatically acquired the MS spectra, and we searched them against the pesticide library, and we got good fit values above 90%, and that was helping us to increase our confidence of identification before reporting the data. And here's just another example where we analyzed an orange sample and then did library searching, and here we found a higher value for imazalil, a typical fungicide which is sprayed to the surface of oranges and other citric fruits. And we did the data processing here in our library view software. That's a very intuitive software interface for library searching and reporting of library search data. All right, and that will bring me to the last topic of my introduction. One of the biggest challenges in food analysis is something what is called matrix effects. Typically ion suppression, but also ion enhancement can be seen. And these ion suppression effects can have a huge effect on the quality of our quantitative results. And for that reason, I think evaluating matrix effects during method development is a very important step to make sure that your method is solid. And here's an experiment explained, which is typically used to evaluate matrix effects. you would infuse a compound of your choice, most likely an internal standard should be used. You infuse that compound post-column directly into the ion source of the mass spectrometer, and that is then generating like a higher signal of that particular internal standard. And then you inject a matrix sample with your autosampler on column. Matrix components will get separated over your column, and then they reach the ion source together with the internal standard. And every time when large amounts of matrix components coallude with the internal standard, you can see the signal dropping, and that's just a measure for ion suppression effect. And one very important tool to minimize or maybe even eliminate ion suppression effects is dilution. I mentioned that earlier already, that the catcher's extracts are typically diluted. Dilution reduces ion suppression effects, and then we have to find typically the right dilution factor best matching our matrices to ensure that we do not have too much matrix effects to report incorrect quantitative data. That's how it could look like. Here we did a matrix effect study analyzing peppermint T samples and T samples. T is one matrix which is very dirty and typically produces lots of ion suppression effects. We started with a dilution of 10x, and then we compared our analysis to results obtained from larger dilution factors. And when we dilute 10 times and 20 times, you would expect that your signal is dropping to 50%, and that would be the red signal, which is like a simulated signal. But in fact, our real signal we observed was significantly higher, and that is just because we diluted our matrix effects by adding the additional dilution step, and we came closer to the real value we quantified. And here on this slide we have 4 different compounds which have been spiked to a T extract, and we analyzed the sample using different dilution factors. And you can see that matrix effects are compound dependent because compounds eluting at different retention times, they experience different matrix effects. And because of that, we also need different dilution factors to completely compensate for matrix effects. And you can see that for these compounds in the T sample, dilution factors of 50 to 200 were necessary to get matrix effects under control. And that is of course an example for a very dirty matrix. If you analyze easier samples, like for instance fruits and vegetables, most likely dilution factors of 5 to 10 are sufficient. But if the samples are more on the dirtier side, like the tea samples here, or maybe spices, or later the feed samples you will hear about, then larger dilution can be very beneficial. So, thank you for listening so far. With this, I would like to hand it over to John.
Very good. Thank you, Andre. Welcome to a joint presentation of Eurofins Cal and AB SCIEX for a brief summary of some of the applications that we're using in our lab for the analysis of animal feeds and feed ingredients. Cal has been a SCIEX customer since 2007 and has been a technology partner with SCIEX since 2012. We have seen AB SCIEX instrumentation as a great vehicle for solving today's analytical challenges in the food and feed markets. The inset photo on this page is the new home of our 40,000 square foot laboratory under construction in the North Technology Park of the University of New Orleans. This laboratory will offer analytical services for the agricultural export industry here in the Port of New Orleans and a wide variety of food and feed contaminant testing for those markets. Eurofins is a growing worldwide laboratory network featuring excellence in what we call bioanalytical testing. It has revenues of $1.5 billion U.S. and over 15,000 employees in 150 laboratories over countries in Europe, the Americas, and in Asia. Over 100,000 different analytical methods are in place in these laboratories, and these laboratories have in-depth industry experience in technical and regulatory matters. Recent acquisition and expansion in North America include Lancaster Laboratories in Pennsylvania, Frontier Global Sciences in Seattle, and a very prominent national water testing lab in Eaton Analytical in Los Angeles. A recent acquisition in South America includes the prominent food and feed lab GCL in Santiago, Chile. Eurofins has built a strong international network of world-class laboratories. The growth of Eurofins' footprint around the world is very apparent. Europe has established food and feed laboratories in all major European countries and expanded rapidly in the Americas and Asia-Pacific regions. Asia and North America will continue to be high-growth areas for Eurofins in coming years, assisting all segments of food and feed production and trade with quality analytical data. In today's CRO environment, a leading laboratory must have the necessary state-of-the-art instrumentation and capacity for high-volume, fast-throughput testing to absorb the peaks and valleys of sample flow. In addition, equipment capacity reserved for analytical method development is quite important to allow improvements and new analytical methods be brought quickly to the market in response to emerging food crises. Eurofins' parallel multi-laboratory capacity for many tests allows flexibility in adapting to the evolving food and feed market needs. So you can see from this list, this list may be a little outdated, but give you an indication of the amount of instrumentation under roof at Eurofins laboratories around the world. Eurofins is commonly operating regional labs in a model called competency centers for specific analytical capability, whether it be chemistry, microbiology, molecular biology, and so forth. This allows concentrated expertise for today's analytical challenges in one location. You should ask yourself, are you sure that your laboratory detects all the contaminants of concern in today's markets at the lowest detection limits, and does it include difficult matrices? Eurofins Cal, located in New Orleans, which is the largest agricultural port in the world, has been a leading laboratory servicing the complex evolving needs of America's agricultural exports industry. The need for rapid and reliable methods for pesticides, veterinary drug residues, and mycotoxins is a continuing challenge for our laboratory. And you see also some of the other competency centers around the world for the various segments of analytical testing that we use to service these markets. So for today's presentation, we have decided on three very important parts of feed analysis that we believe LCMS to have a very clear outlook in these areas. Not included in this discussion today are emerging or unknown non-targeted contaminants, which are reserved for another discussion in itself. But the first part of the discussion will concentrate on multi-residue mycotoxin methods, which we see continuing to evolve, and of course with the use of more labeled internal standards. Medicated feed assays, use of LCMS or LCM SMS methods are evolving in these work areas. And then residues of antibiotics or veterinary drug residues in feed ingredients are also becoming more and more an important area for protecting animals and protecting humans from carryover of medicated product into feed ingredients. So the advantages of modern high-sensitivity triple-quad LCM SMS instrumentation is becoming very clear in the arena of mycotoxin testing. The advantages that we see so far in our experience is, number one, the high sensitivity of current instrumentation is so far well in advance of the current government recommended levels for safe feeds and feed ingredients. So that allows full toxin set HPLC methods that can reduce the runtime typically to 10 to 15 minutes, increasing the throughput and cost efficiency. HPLC is also utilized currently, but LCM SMS procedures are definitely evolving and becoming more of a staple for this type of work. Another advantage, obviously, is reduced interferences sometimes encountered with ELISA or conventional HPLC methods. The increased sensitivity of LCM SMS instruments these days allow for increased dilutions of complex feed samples, still allowing determination of these compounds well below regulatory levels. Trends that we're currently observing is the use of internal standards, which are becoming more and more available for the various mycotoxin compounds. These are labeled internal standards, and the common mycotoxins, of course, are the ones that we typically see in cereal products and cereal grain byproducts now. We also see methods evolving more toward multitoxin analysis with as little as 1 or 2 extraction modes, and we'll be talking about those in just a minute. So we're going to turn this over briefly to Lauren Bailey again for an audience poll.
Thanks, John. Before we get to the audience poll, I just want to remind everyone in the audience that if you do have questions for our speakers during the presentation, you'll see the question box at the top left of your screen when we return to the presentation, and you can enter questions at any time. So for our next audience poll, the question is, are current mycotoxin testing methods by LCM SMS meeting the needs of the global feed trade? And the choices are yes, LCM SMS is the best way to test for mycotoxins. No, LCM SMS still has some limitations that must be overcome. Or maybe LCM SMS is good, but other techniques such as ELISA and HPLC still meet the needs. So are current mycotoxin testing methods by LCM SMS meeting the needs of the global feed trade? And we can see from the results that the majority of you say yes, LCM SMS is the best way to test for mycotoxins, although there is a little mix between some of you saying no, that LCMS has limitations, and that traditional techniques are still working well. So I'll hand this back over to John to see if you have any comments on this.
This is a little surprising in that there's a fairly large segment of people that have doubts about LCM SMS. We have seen some limitations for LCM SMS in spices, but not so much for global feed. Although, by and large, the global feed trade does not work through this laboratory. It's our experience that most of the analytical testing that we do here seems to meet the needs of the global feed trade that's processed through the Port of New Orleans.
Great. Thanks, John, and thanks to the audience for submitting your answer.
So continuing on with mycotoxin testing, one of the first applications of LCM SMS technology for mycotoxin testing was published by a Eurofins scientist, Scarlett Pacelli, in 2005, using SCIEX API 200 instrument for a select group of trichothecenes. With the use of internal standards, Scarlett was able to produce very acceptable data for a series of cereal products. And in the table you see for this particular method was set up with a cleanup using an acetonitrile water extraction and aroma mycocept 226 column, which is still available today and used in our laboratory, followed by LCM SMS analysis on an instrument that is a very nice instrument, but a little bit outdated at this point in time, almost 10 years old now. current instrumentation would likely produce data very similar to this in terms of recovery, but probably a much lower calibration and analytical range. These ranges are not bad actually for the industry. A recent method of note that has been published is the Romer multitoxin method, which was published in 2012. Romer Labs released a method for the 6 major cereal mycotoxins using simple extractions and LCM SMS completion or determination. The method used a series of internal standards to adjust the matrix suppression in a variety of samples that the method was intended for. And as you can see, the preparation flow chart is very simple and rapid, allowing the use of this method in process control as well as export laboratories where speed of testing is very essential. The flow chart is basically a 5- to 6-step flow chart. Calibration in the method is achieved with 7-point standards, including internal standards, but those numbers can be used and modified in each particular laboratory where it's used. Ionization mode was both positive and negative for the various mycotoxins under test. Some more data on that method, on that same method. The LOQs that are shown in the table were generated with the SCIEX 3200 system. LOQs on the order of maybe 5 to 10 lower would probably be achieved on the more modern SCIEX instruments, such as the 5500 instrument that we have in our lab, and data that we produce seems to bear fact that these numbers can be improved significantly with 5500 instrumentation. The column that is used for analytical separation is a Gemini, which is a very popular column these days, and multiphases are very common for use in these types of applications and so forth. And as you can see, all the major mycotoxins are covered with this screen. This is a chromatogram that we produced in our laboratory with very similar conditions, only we used a Phenomenex Luna C18 UPLC column with a Security Guard C18 column. The analytes are listed here. All of these can be done in a single run. And the chromatography is quite nice. ECAL uses targeted mycotoxin methods in our laboratory for the feed types that we encounter and the feed ingredients that we encounter. We feel that a combination of immunoaffinity type column cleanups and chemical type cleanups are the most appropriate. So we don't do multi-class mycotoxins in our laboratory. We separate them into 3 major mycotoxin classes. The first being aflatoxin B1, G1, B2, G2, and ochratoxin using the VICAM afla-ochra immunoaffinity column, followed by LCM SMS. And the data for that is given here in the particular matrices and versus in difficult matrices. Recoveries can vary between 50 to 85% with measurement uncertainties of approximately 10% at the 20 ppb level. And it's going to depend on the particular feed ingredient or feed that we're testing. Matrix match standards are obviously very, very useful in this type of analysis, as we do not use internal standards for the particular mycotoxins as it's done in the Romer method. An example chromatogram of the aflatoxin series and ochratoxin is given here. As you see, very nice sharp peaks in this column, as well as good sensitivity at these levels. The next targeted method is fumonisin. In this case, we're using the VICAM kit, again, the Fumonitest kit with LCM SMS completion. Recoveries are very good at the 100 ppb level, 90% plus for most samples that we test, with 15% measurement uncertainty, which sharply could decrease as the immunity affinity column capacity is approached, which is 7,000 nanograms of analyte. Need to keep this in mind recommend when using immunoaffinity columns that there is an upper limit to their capacity. Instrument conditions here, AB SCIEX 4000, again, the Phenomenex Luna C18 columns would be used for all of the cereal mycotoxins. The final mycotoxin method that we use uses the Romer Multiseptic 26 column, and again, very sensitive. Here's a chromatograph of that series of analytes. Again, on a 4000, recoveries range from 80 to 100%, with measurements RTs less than 10% on this particular. These are very important in feed ingredients, particularly when corn is used as a feed ingredient or corn co-products such as DDG or CGFP. So that brings us to medicated feeds. The use of LCMS or LCMS methods for medicated feed assaying is a relatively unexplored application in the industries now, especially compared to modern tissue residue methods. However, there are some very distinct advantages that we and other labs have found in our laboratory for the use of LCM SMS. The methods would expect to have use for both feed control and verification of data produced by other methods such as microbiological methods or simple HPLC methods. The advantages being that the detection of certain analytes is possible where HPLC methods are not feasible because of the lack of a chromophore or the lack of a particular detector that renders that analysis feasible. The sensitivity and selectivity is suitable for type B and type C medications, allowing dilutions that minimize matrix interferences. The calibration stability of SCIEX instruments is very well known. These instruments are very rugged and capable of meeting the requirements of the medicated feed industry. Trends that we see for medicated feeds is the use of matrix match calibration standards are almost essential for modern feed testing, particularly type C medications, and those are the most commonly used in the development of new methods. The other trend is that we still note that government agencies are a little bit reluctant to accept these methods because these instruments are not available in all the feed testing labs that are used for feed control. So that's still a hurdle that we'll expect to be ultimately resolved, because these methods are, in our opinion, superior over commonly used microbiological or HPLC methods. So how do we create methods for medicated feed assays? There's a couple of different approaches that we use. They're relatively conservative, and the industry we know is conservative with respect to selection of methods. But the main approaches that we've used is, number one, is to adapt an existing LC, HPLC, or single quad LCMS method to improve the sensitivity selectivity of an analyte in a common feed. And we have an example of that in the following slides that we'll talk about. Another approach, obviously, is the creation of an absolutely new LCM SMS method where there's no existing HPLC method. And that's a little bit more challenging, obviously, although the sensitivity of the newer instruments would likely allow fairly simple methods to be developed using standard simple extraction and little to no cleanups. So we recommend, when the creation of new LCMS methods, that partnerships with column technology companies such as Phenomenex, mass spectrometry manufacturers such as AB SCIEX, and even universities to assist with the development or the basic development of methods where existing HPLC methods are not available, and that's what we have done here at Cal as well. So an example of a conversion of an existing HPLC method to LCMS is the analysis of robenidine in feed. The existing method was the EC method L329/54, which we've used in our laboratory to analyze a series of feeds for the poultry industry. In this case, very simple extraction, cleanup with a molecular sieve and alumina, activated alumina, depletion and methanol analysis by LCM SMS. Calibration curve looks quite quite nice. There was a little bit of a deviation at the 1 ppb level, but otherwise excellent linearity for this in the 1 to 50 microgram per liter range, which is relatively typical for this instrument type. It's an API 4000 instrument, and we find the best linearity for these assays to be when the preparation is made in the 1 to 50 microgram per liter range. And here's an example of the chromatography. The chromatography was done on a Kinetics column with an AB SCIEX 4000. On the left-hand side is our lowest calibration level, 1 for the analysis. It's a 4-point calibration, and that is the 1 part per billion standard, which you see excellent sensitivity for. And on the right-hand side is an example of the extracted and cleaned up poultry broiler feed at the 20 to 30 grams per ton range. You see excellent sensitivity and very low background noise for that particular feed. The next example of another adaptation was an example of an LCMS method adapted to LCMS SMS. In this case, a published method in the Journal of AOAC in 2001 by Turnip Seed and Company for the determination of ionophores in animal feeds, or a select list of ionophores there. We used essentially the same extraction and cleanup method that they used in that paper, and we adapted that to LCM SMS with good results. In this case, it was a very straightforward cleanup. Most laboratories can achieve this in a very short time period in their laboratories. Again, the chromatography was very good with very little interferences or noise. Lacillocid is shown in the feed prep on the left-hand chromatogram with both the Q1 and Q2 peaks showing, Q1 and Q2 transitions showing, and the lowest calibration standard in this particular calibration level 1 is shown on the right with slight noise showing in the chromatogram, but obviously a very clean and well and good symmetry chromatogram there. So as a final remark, we intend to continue to use LCMS SMS methods for improving or furthering the use of our technique, improving our techniques for the analysis of active ingredients in medicated feeds. The next topic is another very, very important area these days that we're seeing a lot of interest in, and that is the determination of low-level drug residues in feeds that are used in industries that are highly sensitive to the presence of low-level veterinary drugs. The advantages of this technique are, again, it's a quantitative standard now, LCMS SMS, considered for veterinary drug residues. The new HPLC column phases now allow for a large, wide variety of drug residues, and you'll see that in the example that we present further on in the seminar. So in a single analytical run, multi-classes of drugs can be analyzed with a single HPLC run with very good sensitivity and symmetry of peaks. The newer SCIEX instruments permit positive-negative mode switching, and we have observed in our laboratory that this is very feasible, with both positive and negative modes being tested in the same analytical run with good sensitivity and resolution. So the trends we see is that the more sensitive instrumentation that SCIEX provides is now required to meet a lot of the more current industry needs. So the 5500, 6500 series are going to be critical in meeting the current industry needs. Matrix match standards, obviously, or labeled internal standards. In most cases, matrix match standards are the preferred way to calibrate for these low-level drugs and feeds. Testing for these compounds is normally done in the commercial level at the ingredient purchase or sale. And then the newer cleanup methods that we employ here use strata or HLB column cleanups that have improved recoveries for samples that have been historically very difficult to clean. one method that appeared in the literature last year, which was important. We saw probably one of the first analytical methods for the determination of a single class of drugs in feed was one published in the Journal of AOAC. In this particular case, there was a simple extraction with essentially no cleanup, followed by LCM SMS for the 6 ionophores noted here. Recoveries were not bad. LOQs were a little bit high for the types of industries that are now being served, but the target levels are of toxic significance. So this was a very good and very significant paper that was presented. Another effort that was conducted here at our lab last year was a method that we developed for the salmon industry, and it's the determination of drug residues in poultry meal. Poultry meal is an important ingredient in salmon feed and is required to be tested for antibiotic residues before it enters into that industry as required by the Chilean government. There are extraordinary needs in this industry, and that the LOQs required are typically much lower than the typical target levels that we see in commercial feeds. So it goes way beyond or well below toxicological importance, and now there is more concern about transfer of these residues into the final salmon product from the feed. We have found that the 5500 triple quad is absolutely essential and required to be able to meet the target levels for these specifications, and that a very important part of this is the development of a suitable cleanup procedure. It took us quite a bit of time last year to develop a really good, rugged, and valid method that worked for these types of samples. Poultry meal is quite a complicated matrix, and it has some water solubility, so that the cleanup of the final extract was very important. We were able to achieve LOQs of 5 to 50 ppb in these matrices. Most of the LOQs were at the 20 ppb level, which is very good for this type of matrix. Recoveries with matrix match standards typically are in the 70 to 110% range. Flow chart of this method is given here. It is a relatively simple method. It does involve an evaporation, which does slow the prep down. Typically, we can get this prep done in about 6 hours in our laboratory. Chromatogram and the compound list for that particular application. As you can see, compound classes, the mectins, the phenocols, tetracyclines, fluoroquinolones and quinolones, erythromycin, and some odds and ends here of different types of analytes. We were able to achieve 3 ppb of chloramphenicol, which was important to the industry. And of course, the tetracyclines and the fluoroquinolones were also very important. Tetracyclines have some significance in that they are potential occurrences in poultry meal, so we have to have a good solid method and reproducible method to detect those in poultry meal. An example of an extracted ion chromatogram of N-ofloxacin, very, very sensitive peak on this instrumentation, and the instrument conditions configuration here. We're commonly using Shimadzu HPLC instruments now, and again, we use a pre-column in order to more or less merge the phase that the prep is done into the mobile phase of the system. The analytical column is actually a Phenomenex Gemini column that we developed for this method in conjunction with some of Phenomenex's scientists, and that method was very, very nice, and we're very happy with the work that Phenomenex did with us last year on this prep. And of course, the final instrument is the AB SCIEX 5500 triple quad. Same thing, one of the more sensitive compounds is emamectin, and see the very nice symmetrical peak for emamectin in this thing, very, very highly sensitive peak. I think this is a 0.5 ppb analytical standard. Here are a list of some other applications that we have developed in our laboratory for different preparations and different instrumental conditions for some of the other classes of veterinary drug residues. Clearly, coccidiostats are a risk element in poultry, and a list of the coccidiostats that we do in our laboratory are listed here with their LOQs. These all meet the requirements. These LOQs all meet the requirements of our customers. The dyes, another analytical method, and you can see the extremely low LOQs for this particular application of feed are lower than we've ever seen before in any type of matrix. This particular method took a lot of work in our laboratory last year, but we were finally able to meet MQLs, and again, only because of the use of the 5500 were we able to meet these MQLs. And finally, another challenging class of analytes, the nitrofuran metabolites in a feed matrix, in a dry feed matrix particularly. The low MQLs are shown here. This was another very, very challenging application for us to master, but I'm proud to say that we have the ability to do that now in our laboratory, and we're doing these routinely. So finally, in conclusion, we believe that the analysis of these multi-class drug residues in commercial feed preparations and ingredients by the SCIEX 5500 LCMS instrumentation is both feasible and reliable for today's market needs. The procedure or the method that we developed here is going to be presented next week at another Eurofins SCIEX webinar on June 13th. So if you're interested in some of the details and the challenges that we went through to develop that method, be sure to participate in that webinar. I think you'll find that to be very, very useful. And thank you for your participation today. With that, I'll turn it back over to Lauren.
Thanks, John, and thanks, Andre, for those really nice presentations. We're just about out of time, but I think maybe time for one quick question. And so the question is, what developments are there in analyzing amino acids in feeds using LCM SMS systems? John, do you want to take that one?
We have not done any serious amount of work here on amino acids in feeds. We believe that the high level of amino acids in feeds would probably still be best served with the current methodology. Where we may see some applications is the presence of amino acids in fluids, maybe animal fluids, or in very, very low level matrices where amino acids are not encountered at high levels. Some of our customers have asked us to verify the presence or absence of protein at low levels in their products, since their products are not proteinaceous products, and they've asked us to develop amino acid methods possibly as a measure of trace amounts of proteins in products where proteins are not expected to occur. But we don't currently see LCMS as really being a very big vehicle for the analysis of feeds, unless there are particular amino acids that occur at low levels that are of interest.
Okay, thanks. Andre, did you have anything that you would want to add about the analysis of amino acids using LCM SMS?
Yeah, we did some work on amino acid analysis in all different kind of like matrices. But like John said, we found it more appropriate for body fluids. It's quite popular in clinical laboratories, and we used a specific derivatization technique based on iTrack to enhance quantitative performance. But I would agree for food analysis, especially dirty feed samples, there needs to be some more work done before that becomes routine.
Great, thanks.